All right, welcome back, everybody. I hope that you had excellent, highly opinionated discussions during the coffee break. I had two people come up to me and ask me questions about things that Leonard had said, so I can only imagine the conversations that Leonard must have had during the break. Welcome back. We're going to come back to the session and get started with Kristen Krumhardt, who is a postdoc at NCAR.
So thanks everybody for coming to my talk. I'll be talking about the multifaceted response of coccolithophores to increasing CO2. I'll show you some recent observations of coccolithophore change, and then some modeling results to help explain what we're seeing. So first, shoot. So coccolithophores have a unique influence on the global carbon cycle and biogeochemistry by way of their ability to do both calcification and photosynthesis. The process of calcification actually leads to an increase in CO2, whereas the process of photosynthesis leads to a decrease. And so the balance between these two processes in coccolithophores is important for something like air-sea gas exchange of CO2. And then also, coccolithophores, with their calcium carbonate shell, have a ballasting effect on carbon export to the deep sea. So coccolithophore cells, aggregates containing coccoliths, and fecal pellets of zooplankton having consumed coccolithophores are going to sink faster and ballast organic carbon more efficiently to the deep sea. And so what I'm exploring mainly with this presentation is what is the effect of anthropogenic CO2 inundation into the upper water column. How is that going to affect both the calcification and the photosynthesis of coccolithophores? And so first I start off by showing you some recent observations of coccolithophore changes. And so the first study I wanted to touch on was this one by Natalie Freeman and Nikki Lewandowski, where they observed decreasing calcification in the Southern Ocean, and this is based on particulate inorganic carbon, or PIC, estimates from satellites, where these are converted to calcification rates using an algorithm. And on the left-hand side here, we have this plot of calcification rate versus the year, and this is over the satellite record. For the entire Southern Ocean. And so what we see is for the basin as a whole is that the calcification rate is decreasing. But when we look at the more geographical overview, we do see predominantly negative trends in calcification, as shown by the blue areas on this map. But there are some areas of increased calcification, suggesting that the relationship between ocean acidification and calcification isn't exactly straightforward. And so indeed, if we look at some other studies, now we're going to the North Atlantic. This study by Sarah Varro-Calle and co-authors documented an increase in coccolithophore abundance in the North Atlantic. And so these authors used continuous plankton recorder estimates. And so this is a mesh that filters the plankton from the water column on ships of opportunity, and what they saw is that from 1960 to the 2000s, we see this increase in coccolithophores occurring in the CPR mesh. And so in order to explain this, the authors did some statistical analyses and concluded that the most likely reason for this increase is the additional CO2 coming into the water column from anthropogenic sources. And in order to explain this, we see that on this plot here of growth rate of coccolithophores versus PCO2, we can see that coccolithophores might be stimulated by additional CO2 in the water column. And so this helps to explain as to why we might see an increase in coccolithophores in this region. And so this study is actually from the subpolar North Atlantic regions, as shown as the boat crossings on this map. But this effect was also seen in the subtropical North Atlantic. So this is a study that co-authors and I put out just afterwards, documenting an apparent increase in coccolithophore abundance in the subtropical North Atlantic. And so this is using data from the Bermuda Atlantic time series site. And so here we have coccolithophore pigments, or the chlorophyll A from coccolithophores. And over the period from the late 80s to 2014, and we're seeing an overall increase in these coccolithophore pigments over time. And actually, they're also positively correlated with the dissolved inorganic carbon in the water, and especially the bicarbonate fraction of this. And this is in contrast to total chlorophyll. So the rest of the phytoplankton community doesn't seem to be responding as positively to the additional DIC as coccolithophores are. And so this is just more evidence that perhaps coccolithophores are getting this carbon fertilization effect on their photosynthesis from the increasing anthropogenic carbon. And this was just further corroborated by satellite-derived particulate inorganic carbon. So on these maps, I have the CWIFS era trends in particulate inorganic carbon on the left-hand side, and then the trends from the MODIS satellite on the right-hand side. All the trends are plotted on the top maps, and then just the significant trends on the bottom maps. And what we see is predominantly positive trends in particulate inorganic carbon in this region, just more evidence that coccolithophores appear to be increasing in abundance in the North Atlantic. And then one last observational study that I wanted to touch on was this one by Winter et al. From 2013. And they document a poleward expansion of the coccolithophore species Emmeliana huxleyi. And this is from using both satellite observations as well as in situ observations. And so this map right here in the background with the white area, that's the bloom extent of coccolithophores from the CWIFS era. And then the red outline showed the previous satellite observations of coccolithophore maximum bloom extent from 1978 to 1986. So what we can see is that this species of coccolithophore has appeared to expand its range, especially in the poleward direction. And this is also corroborated by in situ data. So if we just focus on the red line on this plot, we have the concentration of coccolithophores in the water column. And what we can see is that in this particular sector of the Southern Ocean, they appear to be increasing as well. So overall, within observations, we have a decrease in calcification in the Southern Ocean overall, increases in the North Atlantic in coccolithophores, and also this poleward expansion of this particular coccolithophore species. And so to better understand what's going on here, we can use an Earth system model to help parse apart the different effects. So for that, I'm using the Community Earth System Model, or CSM. And this depicts the standard configuration for CSM, where we have small phytoplankton, which is a rather diverse group of different phytoplankton. And then we have the diatoms, those silicifiers, the diazotrophs, the infixers, and one zooplankton type that grazes all of these phytoplankton functional types. And so my job basically was to pull out this implicit fraction of calcifiers from the small phytoplankton group that was representative of coccolithophores, and put them into their own phytoplankton functional type that is sensitive in both growth and calcification to changing CO2. And when making this phytoplankton functional type, I wanted to make it as representative of as many coccolithophores species as possible. So for that, I did this big meta-analysis of pulling together as much physiological data I could with relating changing environmental conditions to their growth and their calcification. And since this talk is focusing most on changing carbon dioxide levels, I'll go into more detail on that meta-analysis. So there's a lot of data on this plot, but I'll break it down for you. So on this top plot, we have carbon-specific growth rate on the y-axis as a function of particulate inorganic carbon. And on the right-hand side of the plot, I've broken up the data into different species, or Emmeliana huxleyi morphotypes. And what you can see is that, in general, there's a lot of interspecific variability among different kinds of coccolithophores. But when we pull all the data together, we can fit a Michaelis-Menten uptake kinetics curve to this line and derive a half saturation constant for CO2. So that's that I put into the model. And then just going to the bottom plot, now this is the ratio of calcium carbonate to organic carbon in the Coccolithophore biomass. And this is kind of a measure of calcification. So on the right-hand side, again, I've broken it up based on different species, and some species just tend to calcify more than others. But when we gather all this data together, because remember, I want to make this as representative of as many Coccolithophore species as possible, then you get this plot on the left-hand side, and there is a lot of scatter, but overall we're able to fit a significant linear trend between this calcium carbonate to organic carbon ratio and as a function of CO2. So overall we see this as a negative relationship. Now this is put into the model. And when you multiply this line times this one, you get the calcification rate, and this is going to look a lot like what Leonard showed with a kind of an optimum calcification range of PCO2. And so in this talk, I'll refer to this top part as the carbon fertilization effect on growth. So more carbon, their growth may be stimulated. And then this bottom one is more the ocean acidification effect on the calcification. And so I put these into the model, and this is a summary of all the relationships I put into the model between Coccolithophore growth and calcification. And what we have is these two plots basically just summarize what I showed you on the previous graph, so the carbon fertilization effect and the ocean acidification effect on calcification. And then if we look to temperature, basically a power function is determining the temperature-dependent maximum growth rate. This is in contrast to the other phytoplankton functional types in the model, which use a Q10 curve. And then this was shown to best fit the observations or the data from physiological studies for Coccolithophores. And then also, Coccolithophores tend to calcify less under really low temperatures. So this was put into the model as a linear decrease in calcification for temperatures lower than 11 degrees Celsius. And then with the relationship with nutrients, with growth rate, so Coccolithophores tend to have efficient nutrient uptake kinetics. And for instance, they can outcompete diatoms when nutrients become limiting. And so this was put into the model as efficient half saturation constants for nutrient uptake. And then one of the things that I found rather interesting about this meta-analysis that I did was that as nutrients tend to become more scarce, then this calcification actually increases. So their growth rate slows down, but they continue to calcify. And so this is in the model as a negative relationship between nutrient limitation and this calcium carbonate to organic carbon ratio in Coccolithophores. So once I had these relationships put into the model, the next step is validation, and I have mini validations in the paper where this is published, but for now I'll just show you the comparison to PIC from satellite. And so this is PIC from satellite there on the top, and then the top 50-meter average Coccolithophore calcium carbonate in the model. And overall, we tend to match the patterns, although the blooms in the North Pacific and North Atlantic tend to be a bit too far south. We also get the suggestion of a great calcite belt, although it seems to be more abundant in certain regions. And then we're on the right order of magnitude for the subtropics and the tropics. So Jen, just to briefly describe my experimental setup. So here we're just testing the effects of increasing CO2. So these are ocean sea ice only runs, run under normal year climate. So the same climatology repeating over and over again. They're run on the nominal 1 degree resolution for CSM. They're started with an ocean physics spin-up, then a pre-industrial control run, and then we just branched off higher CO2 runs and compare them after the models come into equilibrium. And again, I want to emphasize that there's no warming effect in these runs. We're just looking at the effect of CO2. And so these maps show the depth integrated calcium carbonate production in the model. So this is under pre-industrial CO2, and this is 400 ppm CO2 or modern day. And here's a difference map or a percent change map to show how these maps are different. And so basically what we see going from pre-industrial to 400 ppm CO2 is a slight reduction in calcification in most areas of the ocean, but then we do see these stark increases in certain regions, and those are places where the coccolithophores are feeling this carbon fertilization effect. They're better able to compete with the other phytoplankton functional types in the model, and so calcification increases there dramatically, and this results in a 6% increase relative to in globally integrated calcification relative to pre-industrial times. But then if we go to 900 ppm CO2, we get a much stronger ocean acidification effect, and now this outweighs the carbon fertilization effect, and we get a negative change relative to pre-industrial. And then looking at, this is just another way to look at that. This is the globally net integrated calcium carbonate production in the model. The pre-industrial control is in the black, and then we can see that this is the increase from pre-industrial to 400 ppm CO2, while this is the decrease going to 900 ppm CO2. And if we look at the green line here, this is at 600 ppm CO2, and that shows that that's kind of where the positive effects of the carbon fertilization are balancing the ocean acidification effects. And so if we look at some regional results from these model runs, you get, so these are from the North Atlantic, from the subpolar biome on the top, and these are in the depth versus time space, and the colors are showing the coccolithophore biomass. This is the last 10 years of the pre-industrial control, followed by the first 10 years of the 400 ppm CO2 run. And what we can see is that in both the subpolar biome, shown in orange, and the subtropical seasonally stratified biome in purple, we see this shift to a higher coccolithophore biomass as soon as the additional carbon is getting into the water column. So this is more that carbon fertilization effect. And this is, again, similar to the observations I mentioned earlier. And then if we look at the Southern Ocean, now this is the same map I showed you earlier from Freeman and Lewandowski, showing the negative trends in calcification in the Southern Ocean with some positive trends in there too. And if we look at the same change from the model, going from pre-industrial CO2 to 400 ppm CO2, we see that predominantly in the Southern Ocean there's negative trend or negative change in calcification, but there are some regions again that are experiencing that carbon fertilization effect, but these are outweighed by the ocean acidification effect, which is shown on this plot of the total calcium carbonate production in the subpolar Southern Ocean biome. Pre-industrial is in black, and then this 400 ppm is branched up here, showing a lower total calcification for the Southern Ocean. And then as CO2 increases, you get more or less calcification here. And so just to summarize the results of these CO2 experiments, so these are, I've shown you the explicit coccolithophore parameterization in CSM, and this was based on physiological experiments that showed this carbon fertilization effect. So as CO2 increases, their growth rate can increase, and the photosynthesis can increase. And then, but there's also a decreased calcification from ocean acidification. The changes we see in the model are supported by the recent observations of coccolithophore changes. And then overall, increasing CO2 results in more coccolithophores just in certain regions, but all of them have lower calcium carbonate to organic carbon ratios in their biomass. And there are some biogeochemical implications of these findings, such as places that have increased coccolithophore abundance have increased E ratio or increased export of organic carbon. And also places like most areas of the ocean saw this decreased calcification. So these areas would have increased carbon dioxide uptake. And I just wanted to point out some of the limitations of this model study though. So far I've just tested the effects of increasing CO2, and so far there's no warming in these simulations, so warming may actually complicate these changes. For instance, like increasing sea surface temperatures could increase the calcification and counteract some of the ocean acidification effects. Also, all these simulations were run with just normal year forcing, so repeating climatology. And it would be, and having interannual variability in climate would be more realistic. And right now there's no negative feedback of low calcification, such as increased grazing. So if coccolithophores are more lightly calcified, they might not have so much protection from grazers. So that's something we could potentially add in. And then also there's only phytoplankton calcifier. There's only a coccolithophore in the model as a calcifier. So maybe having a zooplankton calcifier would complicate these changes as well. And with that, I'd like to thank my co-authors on the CSM work and the authors of all the studies that I described. And thanks for your attention.
Questions for Kristen? Ooh, so many hands so quickly.
Chris Sabine, Hawaii. Great talk, thank you very much. So one of the observations we've seen in the accumulation of anthropogenic CO2 in the ocean is that there's been relatively less or a slowdown in anthropogenic uptake in the North Atlantic relative to other areas. Is your model reflecting that, and could that perhaps also help explain some of these differences between the North Atlantic blooms and the Southern Ocean?
I'm not sure it would exactly reflect it because, these are just step changes and it's not like a slow incremental change, although they are coming into equilibrium. So I would, the CO2 in the atmosphere is coming into equilibrium with the water. So it might, in equilibrium, it might actually be reflective of these changes, but I'll have to think about it more and we can discuss later. Thanks.
Hi, Kristen. Barney Balch from Bigelow Lab. Nice talk. Could you comment on the factors that are responsible in the Southern Ocean for turning them from net negative to net positive in the calcification? You showed these charts where parts of the Southern Ocean showed clear cases of increased calcification. What factors do you think would have been responsible for that?
So that in the observational data, you think? Or?
Well, in your model, both you saw.
Okay, well, so in the model, the response is solely due to the increasing CO2, so that's the carbon fertilization effect, so it has to be that. But in the real world, in the observations, it could also be due to increasing temperature. So in one of the regions, there was this. The authors kind of pull this one out, and they attribute that to a poleward migration of the polar front. So, so maybe that was the reason that we saw increases in that region. I mean, it's impossible to know the exact reason, but that's how the authors speculate that the poleward migration is the reason that for that increase. And I don't know much explanation for these ones in the Pacific.
From your model, it looks like on the right there, that the red areas are following the subantarctic front.
Yeah. So I don't know if that would, support what they had assumed is happening there. But
Thanks.
Hi, Stephanie Dukevich, MIT. I guess there's sort of two questions. One.
I don't think the microwave or the microphone is on.
Is that one working? Great. I'm Stephanie Dukevich, MIT. So two questions. One is you only allowed the coccolithophores to have a benefit from CO2 fertilization, but potentially some of the other phytoplankton might have, and that would change some of the competition and therefore change some of these changes. So that was one is if you put that in, is it something you're thinking about, that sort of thing? And the other one also was, did you try and actually run this also without a CO2 fertilization? Because some of those changes could also be just the fact that coccolithophores are probably, you probably have them having a slightly better affinity than diatoms, for instance, and so their southward movement could just be because there's less nutrients in the water.
Oh, right,
so there may be sort of compounding effects there.
Yeah, so first on the question about the other phytoplankton maybe getting a carbon fertilization effect. Well, so far we haven't added that, but the model certainly has the capability to add a half saturation constant for other phytoplankton types. The evidence hasn't been as clear as for coccolithophores, so I haven't added anything about that yet. And then also, in terms of the competition with the other phytoplankton, it's certainly possible that increased nutrient limitation could shift, cause an increase in coccolithophores just by them having better success under nutrient limitation than, say, diatoms. So far in these model results, that's not the case because we're only changing the CO2, but it's certainly with like future warming and increased acidification, that could be definitely another reason for their increase is the competition factor. So more to explore, I guess.